understanding biological functions often requires investigation of the molecular compositions and the dynamics at high spatial and temporal resolution. Here's a schematic showing a subcellular structure called synaptic bouton, which makes up a neuronal connection in our brain. This rendered image contains 60 different synaptic proteins occupying about 7% of its volume and constituting minute structures ranging from nanometers to micrometers in size. When we zoom out, a single neuron can have as many as 30,000 of such synaptic connections and extend millimeters, if not longer, in its full length. When we further zoom out, the human brain weighs about 1.4 kilograms and extends about 15 centimeters in length. It can contain up to 100 billion neurons interconnected with each other. The fundamental question my lab is pursuing is how can we identify and locate specific molecular markers across such a broad length scales and biological complexity. One approach my lab is exploring is a biochemical process that transforms biological specimens so that it can be studied more easily at higher spatial resolution. In this approach, cells and tissues are first fixed and chemically modified with a small molecule reagent. The modified specimens are then polymerized in situ to form a superabsorbent hydrogel composite. During this process, biomolecules of interest are anchored to the hydrogel chains. Finally, after digestion, the gel specimens are expanded in water, causing the anchored molecules to separate from each other. Because of this swelling effect, biological specimens are magnified physically so that a higher spatial resolution can be achieved. When we combine this biochemical process with the latest microscopy techniques, we can capture the native molecular profiles of cells and tissues with nanoscopic resolution. For example, we have performed nanoscale imaging of different neuronal structures using a fluorescence imaging technique called the lattice light sheet microscopy. This includes the investigation of pre- and postsynaptic proteins in the mouse cortex, axons and their myelination profiles in a similar brain region, dopaminergic neurons and their myelination profiles across the entirety of a fly brain, as well as the correlation between axonal degeneration and certain disease markers in the human brain. Thank you so much.